Good morning, children. So we are back again with genetics. The previous class I had explained you Mendel's first law based on monohybrid cross. I have again shown the same character with the monohybrid cross with same characteristic, round and wrinkle. This cross only I had shown in the previous class. Now I will be explaining what was the first law. The first law was law of dominance. Now the second law, this was of course based on, can be explained on monohybrid cross. What have we seen in monohybrid cross? We have in law of dominance, if there is an allele, is it a pair of allele, is it controlling contrasting character, one expresses itself more than the other, the more expressive allele is dominant, whereas the less expressive allele is recessive, is it? So we have done the law of dominance. Now let us come to the second law, and based on this only, the law of segregation. This particular law is also known as the law of purity of gamut. I'll explain why do we call it law of purity of gametes. Now, in this particular law, Mendel stated that the alleles remain in pair. Suppose I show it as round and wrinkle, alleles remain in pair. Is it all right? But they never blend with each other. Sorry, they never blend with each other. Here what I am showing, this is the allele for round and this is allele for wrinkle. Okay, so the members of this allelic pair, they segregate during gamete formation. Repeating once more, the alleles remain in pair. What are alleles? Again I am repeating, alleles are genes, a pair of genes controlling a particular character. Allele is never singular, and we cannot say one allele, means one gene cannot be termed as allele. Alleles are pairs, is it all right? Means controlling a particular character. Say controlling, in this case it is the shape of seed. Is it all right? So what are the genes controlling the shape of seed? So it can be capital R, it can be small r, or it can be capital R, small r. These are similar alleles, these are dissimilar. This is a similar allele, this is dissimilar allele. Is it all right? So whatever, and the laws are better explained in dissimilar alleles, because similar allele we don't understand, both are same. Now he stated that the alleles remain in pair. They always remain in pair. In a diploid organisms, the allele remain in pair. Is it clear? As shown here. Okay. But, and they never blend with each other. They do not contaminate each other. They have their own individuality. The genes, the members of the allele have their own, means they have their individuality, and they do not contaminate each other. They just remain in pair. But they segregate, and they segregate during gamete formation. Is it all right? So that is why it is called the law of segregation. What is it? That means he said, and why did he say? I'll explain. He said the members of an allele, this is allelic pair, the members of an allele, they remain together, they stay together. Is it clear? And they remain in a pair, and they never contaminate each other. They do not blend with each other. What do? They remain together. They just remain together, but they separate during gamete formation. Is it clear? Gamete formation, the members of the alleles which were together in diploid organism, they separate during gamete formation. These two separate. Is it clear? And this is why it is known as law of segregation. Now, why did he say that these? Why did he consider that the allelic pairs are not, the members of the alleles are not blending? Okay, so what has happened? Suppose we consider that the members of the allele are blending. Suppose capital R and small r, what we have shown, instead of, like, let us consider that they have blended. R and R have blended. Is it all right? Now, if blending had happened, let us consider till here, till F1 generation, R is the dominant character and small r is the recessive character. Let us consider even after blending, the dominant character shows itself, so it is round. Till here, we cannot prove that no blending has occurred. Here, till here, we cannot prove that no blending has occurred. May have, may not have. Now, where is it proved that blending has not occurred? In F2 generation. How? In F2 generation, we see that one of the character, the recessive character, shows itself. If blending had taken place, then recessive character would not have come out separately. Blending means mixing, total mixing. Blending, the genes are mixing with each other. Since there is no blending, that is why the recessive character in F2 generation could be shown. If we consider that blending had taken place here, mixing of the genes had taken place here, R, small r, or the recessive character, wrinkle character, would not have come out in F2 generation. But in F2 generation, we are finding a wrinkle character, the recessive character. That shows that the alleles are staying in pair, they remain in pair, but they do not blend or contaminate each other. They do not mix with each other. They simply separate during gamete formation. Is it? So this is what is law of segregation. I'll give you in writing. And now, why is this known as the law of purity of gametes? The reason is, if you see the organism, it can be, if you see the character, it can be capital R, or small r, or capital R, small r. Is it clear? Like, what are the characters? So this is homozygous, this is heterozygous. I have already explained you what is homozygous and what is heterozygous, is it? So these two are homozygous and this is heterozygous. The organism can be homozygous, can have a homozygous character, or can have heterozygous character. But what are the gametes formed by this? Capital R. Let us see. What are the gametes formed? This is forming this. Okay, what is this? This is forming small r. What kind of gamete is this forming? Capital R, small r. Is it all right? Now, what does it prove? It proves that the gametes either has a capital R, that means gene for round, or has, don't see who is, now only concentrate on this part. These are the gametes. Is it clear? Can you see any of the gamete which is mixed? No. This is only having round gene. This is only having round gene. This is only having wrinkle gene. This is wrinkle gene. This is round gene. This is wrinkle gene. Is it clear? So gamete either has a round gene or has a wrinkle gene, either has a tall gene or has a dwarf gene. That means the gamete either had a dominant character or had the dominant gene or had the recessive gene. Is it clear? These gametes are never heterozygous. That is why, since the pair is segregating, since the members of the allelic pair is segregating, gametes are always pure. Is it clear? It has only one kind of gene. That is why this is also known as law of purity of gametes. So how do we define it? You can write the definition what I am giving, or the definition which is given in the book. Anything, only the language is changing. So what is this? Members of allele remain in pair they do not mix. You can use the term mix, blend, contaminate, each other. Contaminating term is given mixing. Is it all right? Contaminate each other. The members segregate. The meaning of segregation is separation. Segregate during gamete formation. This is law of segregation. And why? Hence the gametes formed. Hence the gametes formed are always pure. Is it? The meaning is capital R, this is heterozygous, this is not pure, this is hybrid. The character is, it has a hybrid heterozygous character. Is it all right? But when this heterozygous thing is separating, is it all right? What is it forming? It is forming two gamete. Is it all right? So one is only having round, the other is only having wrinkle. So this is pure round, no other form is here, and this is pure wrinkle. So the gametes which are formed are always pure. That is why this law is also known as law of purity of gametes. So the first two laws of Mendel are given on the basis of monohybrid cross. Now again I'll explain monohybrid cross today. Since that day I had used the term pure breeding plants. Now you will understand what are pure breeding plants. Homozygous. So pure breeding. What is monohybrid cross? Monohybrid cross is a cross between two pure breeding plants. Is it clear? Considering one character at a time. Pure breeding means when we started with it is, suppose we are taking round and wrinkle, or we are taking wrinkle, is it clear, or we are taking tall or dwarf, whatever. Is it clear? This should be, this round, wrinkle is the recessive character is always pure. I told you in the previous class only that recessive character is expressed only when it is in homozygous form. Is it all right? So pure breeding plant means it should be RR. It should not be capital R, small r. This is not pure breeding. Is it clear? This is hybrid. This is hybrid. So you have to, means it is pure breeding plant. Wrinkle should be small r, small r. Is it clear? Tall should be capital T, not capital T, small t. This is not pure breeding. Is it all right? So monohybrid cross is the cross between two pure breeding plants considering one character at a time. So that is what is monohybrid cross. Today we will again, after this I'll start with dihybrid cross. What is a dihybrid cross?